Hello, we have today with us Professor Jerri Bartholomew and Ben Americus from Oregon State University. And we're talking again about this special issue of International Journal for Parasitology, which was specifically focused on fish parasites. And its publication was supposed to coincide with the 10th Conference or International Symposium on Fish Parasites. Unfortunately, because of COVID-19, the symposium has been postponed to next year, but we are still running the special issue, which has been just published, and we're talking to authors of the some papers. And today, as I mentioned, we are joined by Professor Jerri Bartholomew and Ben Americus. Jerri, you have been to quite a few of the ISFP meetings before, and I was wondering if you could share with us some of your best memories.
Okay, well, I think actually the Australian meeting would have been my third ISFP. And I have really enjoyed the meetings that I've been to. Wanted to pull up. Okay, well, one of the things that I've really enjoyed about these meetings has been the opportunity to get together with my fellow mixozoan researchers. And at each of these meetings, we've had a set of mini sessions called the Mixozoan Workshop. And they've usually been 3 or 4 sessions over the course of a day. And of course, included an evening of going out and having some drinks together and talking about things. And it's been a really great way for researchers with common interests from around the world to get together and just talk. And these are some scenes from the Valencia meeting with Ariadna Sichabobadia and I hosting the Mixozoan Workshop, and a couple of photos from the night at the aquarium, which was really pretty special, a dinner in the round with fish watching us eat.
Yeah, that was quite amazing. I fully agree with you.
Absolutely beautiful. Yeah, and my first ISFP meeting was in Viterbo, Italy, which was also a really beautiful location and a great meeting.
Yeah, and I think the organizers really made a huge effort both in Valencia and Viterbo to make their meetings great success. So I remember that your Mixozoan workshops and how many people attend them and the big diversity of researchers from different countries, different ages, everybody really passionate about mixozoans. So why mixozoans? What do you find so exciting about them after all this?
Well, Ben's going to talk a little bit about that, but I've been working on mixozoans back when they were still known as, they weren't even known to be organisms with complex life cycles. So I've seen a lot of changes in this group. I think my first work with mixos started in 1981. And the first life cycle wasn't solved until about 1984 or 86. So it was really a pretty exciting time to work with these group of organisms. I will have to say that being an artist also, I think that visually they have a lot to offer. There's a lot of diversity. They can infect just about any species of fish, and they're found in about any tissue or organ within a fish. So there are thousands of species, and that just offers an endless opportunity for questions.
So going to questions, I think we might move to Ben, because Ben is the first author of the paper in the special issue of IJP. So congratulations, Ben. That's a really fantastic achievement.
Well, thank you. No, happy to be in it.
Yeah. So I can see shirt has some of the pictures. Can you explain to us what's on your shirt?
Oh yeah. This is my friend made this for me. I can zoom in. It's like the summary of the paper. The paper's about these stinging cells and mixozoans, and these are stinging cells. But it's a review paper comparing stinging cells and mixozoans to stinging cells in things like jellyfish. Yes.
Really fascinating, because obviously the taxonomy of mixozoans changed quite a bit, and they were assigned to different groups over time.
Yeah.
It's not a taxonomy exam, so I'm not going to ask you.
Oh, no.
This. I'm more interested how you decided to work on this particular project, and also what do you think are the most important finding of the paper?
Yeah, I guess I started doing mixozoans just as I guess I like come from a fish family. My dad's a fisherman, my brother's a fisherman, so I wanted to do some kind of project on fish. And I started working on mixozoans just as a fish health kind of issue. And yeah, pretty quickly my grant funding source, they directed me to looking into these stinging cells. They're called nematocysts. And this is like really essential to mixozoans attaching to fish and infecting fish. So yeah, this paper in IJP, it's a review comparing stinging cells in mixozoans, these parasites to stinging cells in jellyfish or anemones, because Jerri was part of this work originally, but they've known for probably like, I guess 20 years or so that mixozoans, these weird freshwater things, are actually part of this really diverse family of cnidarians. And yeah, it's fair. I think some of the big takeaways I think in this were looking at kind of the protein composition of mixozoan nematocysts and kind of seeing that they're a lot less complex than these similar organelles in free-living cnidarians.
So you propose some significant changes to terminology about the words to use for different part of mixozoan. So can you explain to us what you think should change?
Yeah, I think this polar capsule was the term people used to describe mixozoans. I think it's it kind of just, my opinion right currently is it detracts from the same commonality. I think people should call polar capsules nematocysts. I think that's the vision, which is a small one, but I think it gets people thinking more about the similarities. Basically, like all of this came from my master's work, and all of my PhD work now is comparing mixozoans to free-living cnidarians. I think if more people apply some of these techniques that we use on anemones and jellyfish to mixozoans, I think there's a lot more we can learn about them. A lot of interesting stuff and a lot of stuff that's relevant to fish diseases.
So your PhD is continuation of what's in the paper? Is that what you're saying?
Yeah. Specifically, in the paper we mentioned, there's a subsection on sensory apparatuses, comparing how a jellyfish senses a fish to how a mixozoan senses a fish. And yeah, my PhD is specifically comparing and contrasting those features.
So the two questions that you posed at the very end of your paper, asking about the sensory apparatus signals discharge, and if they exist, what's the role of any ancestral cnidarian venom compounds in mixozoans? That's really what you are working on in your PhD.
Yep. Yeah, there's should be there's a venom paper coming out hopefully within the next 6 months or so. It's in internal review now, but yeah, venoms and sensing, and yeah, those are the big two I think I'm interested in.
And your paper is really beautifully illustrated. I think it's one of the nicest papers in the whole issue from the point of view of the figures. So did you do all the figures yourself, or other authors contributed?
Yeah, I can share. I have it up on my Twitter, but the eventual figures, I did a bunch of like bad sketches of the. I can share. I can email it you later or something. But basically, I did a bunch of like 6 months of bad sketches of these things. Then my boss, Stephen Atkinson, he was originally a graphic designer and joined Jerri's group to get his PhD, and he did the published figures. But yeah, it was cool. It was cool to see like this was my first paper, so it was cool to see like something go from like a napkin to a nice looking figure. And yeah, Stephen and Jerri were a big part of that.
And you mentioned that you were initially interested in mixozoans as a fish health issue. So do you still think that they are a big fish health issue? And which particular species do they affect?
Yeah, I think so. Yeah, sea shasta. I guess the biggest issue right now is sea shasta. In Oregon that I'm familiar with is sea shasta in relation to the Klamath River and these dams that they're talking about removing. Jerri can, I'm sure, talk more in deep about this, but it's I think so. yeah, when I go to talks and give presentations on this, there's a lot of people that are involved with that dam removal process that are interested in this. And I think, yeah, what keeps why I stayed doing mixozoans was because like I can explain it to my parents, like these things affect fish, they like fish, they make money off of fish, but they're also interesting from a like evolutionary biology perspective.
Mm-hmm. Yeah, that sounds really fascinating, and I would like to congratulate you on the paper again. And I'm really looking forward to your future papers coming from your PhD, because it sounds like hopefully you will solve all these mysteries that you're talking about in the current paper.
Hopefully, yeah, that's your goal, yeah.
So you're comparing particular species of mixozoans and jellyfish, or do you just look across many different species?
Yeah, it's mostly sea shasta, which is Jerry's bread and butter. And we can keep it year round in the lab. Mostly sea shasta to this little freshwater brackish water anemone called the Nematostella vectensis. Those are like the two models we're comparing.
But you're also doing some work with Mixobolus cerebralis too.
Oh wow! So it's all really interesting, and I guess when you get more detailed results, then you can go and compare to other species as well, if anybody wants to look at a even bigger picture, so to say, because you're already obviously looking at a big picture. Sorry, Ben?
Oh, yeah. So we do, even in this paper, as Jerry mentioned, we do include some photos of M. cerebralis. That's kind of like sea shasta and M. cerebralis.
So Jerry, you've been working on mixozoans for quite a while, as you said before. So do you have one favorite one, or are they all your favorite?
Well, I have to claim favoritism for sea shasta just because it is evolutionarily, I think, a really interesting species because it kind of bridges the marine freshwater life cycles by having what we traditionally think of as a marine organism as its host, freshwater polychaetes, which we aren't calling them polychaetes anymore, but they're very rare. I think there are only 11 or 12 species. And they're using a host that's anadromous. So I think it's a neat model because it pulls together some aspects of freshwater mixozoans, but also saltwater mixozoans. And it's also interesting because unlike some parasites like Mixobolus cerebralis that have been virtually shipped all over the world and been able to establish in many places, this hasn't spread outside of the original region where it was described. So it's still only found in the Pacific Northwest, basically. So, it's something that I guess is particular to our region, which makes it, a nice thing to study for us.
And is there any of them most artistically pleasing? Like any of the mixozoans look better than others? Do you have like beautiful mixozoans?
Yeah. Some of the marine Ceratomyxes are really lovely with the long, arching spore forms. The triactinomyxon, or the actinospore stage of mixozoans, is pretty dramatically different than the mixospore stage. So, those just provide a nice contrast. But some of those are really lovely. I think the Sidlichiella, where they form mesh groupings, are really, really pretty.
So you're one of the scientists who manages to put art and science together, and you're an artist and scientist, or scientist and artist, like Leonardo da Vinci style.
I don't know about that.
So what came first? Were you first interested in art or science? And did the way the mixozoans look inspired you to do art?
Well, I think I've always done art in some form, and it's probably been in the last 20 years that I've started to look more closely at using art and science together in different forms. And during that period, I've been working exclusively in glass. So that's the medium that I've been exploring these connections between art and science. But I've also been exploring it by collaborations with other artists and particularly with musicians. And
Wow.
it's been really interesting to
Mhm,
share our science
mhm.
and see how other people interpret it. So this is a talk that I was planning on giving at the meeting this past summer, and which hopefully I'll be giving in some
Next
form.
year, yeah.
Yep, next year. I'll have had quite a bit more time to work on the artwork part of this because I'm on
Mhm,
sabbatical this year, and that's what I'm working on. But I've taken that idea of the nematocysts and the concept that they're kind of these weapons of micro destruction and used them to explore artistically. And this slide shows a collaboration that we have with some Israeli researchers in Tamar Lotan's lab. And this is a drawing by an artist here at Oregon State University. And he's somebody that I've been co-teaching a class on art and science with for the last couple years. So in further exploring that, you know, we just talked a little bit about the diversity of myxozoan forms. This plate shows some of the diversity of myxozoan forms, and you can see why I find them so interesting. Here's one of these net-forming myxozoans and the nicely arched ceratомyxoforms. And they're really quite lovely, even if you aren't an artist. But what I've done is try and take them and look at them a little bit more closely than I have as a scientist and in a different way. And this is a piece from a series that I did a couple of years ago where I took, I etched different myxozoans into glass and really looked at their cellular structures. And this is a series that I'm continuing, and it's going to be called, I think, in the end, Abstracted. And it will go from very literal pieces that pretty much describe a manuscript to further and further abstraction. And what I like about that is that we use the term abstract in both science and art, and it means two entirely different things. But as scientists, our abstracts really encapsulate the whole paper or that whole body of work. And that's, I think, what a good piece of art can do too.
It's really interesting that it's not just visual art, which is obviously really beautiful, but science can be also translated into music. And Jerri will, can you share with us maybe some of the pieces of music that have been composed based on your results?
Okay,
There are stories told from the days of old of a river flowing free and a ruby sea, of salmon bound for a natal ground that they knew from infancy, calls from destiny, as the river and its spirit to the ocean. Now the waters yield to concrete and rock that lift up to the sky, and many years go by that the salmon runs make no display, and the sport that make it through the fray are numbered few. While the people of the land stand in wonder. Klamath Basin River tale, where mysteries are illuminated, revelations made from the innate. Hunt the story to the sand, silt where polychaetes lay hiding, bring them up, here researchers say. It's a crazy life for a centuries ago, with the waters high and the outflow moving slow. There's a plea for aquatic mercy, and to hear a ruling say, let the floodgates go. This is how it goes. Digipala keeps the parasites drift and hold for a breath from the hunted in their rosy folds, a marauding crew. With a pass through the gills, harpoons away, and a strike from nematocysts that pierce membranes, mortal games ensue. As they wiggle into challenge prey defenses. Now a parasite can bear no raid alone, for the multivoltinic ways to hold their own till the rains accrue. And the heat of the summer songs long sustained gives away to the burden of infections reign when the siege is through. And the microbes commandeer a fulsome bounty. The salmon soon to waste away. At the brink of expiration, the restless raiders don a second face. Venture to the riverbed, fickle in their host's selection to plunder sands again for polycates. It's a crazy night. Or a parasite taking out your milk and cocoa, navigable through a river not their own, from centuries ago, with the waters high and the outflow moving slow. There's a need for aquatic mercy, and to hear a ruling say, let the floodgates go.
That's fantastic. Thank you for sharing it with us. I really enjoyed it. That must be like the best hit of fish parasitology.
It was a lot of fun. And each of the musicians has had such a different take on things. This was a very literal and fun. The others have been really just sonifying data and looking for patterns. So
yeah, that's fantastic. We're looking to hearing and seeing more of this when you come next year to the ISFP in Cairns and give your a keynote presentation. And once again, I would like to congratulate Ben and you on the paper in IGP. That's a really interesting paper and changes our understanding of Myxozoa and the position in taxonomy. So that's really fantastic as well. So thank you very much for the interview. I really appreciate you joining us today. And I hope I'll see you in Cairns next year.
So Barbara, thank you so much for this opportunity to talk to our fellow scientists. And I hope that we'll be seeing all of you or many of you next summer in Australia. So thanks again. Ben, do you have anything you want to add?
Yeah, I'll say thank you for putting this highlight on my paper, and yeah, I hope to meet you all in person post-COVID vaccine.
Okay, thanks and bye.